For those of you who don't know me, I'm Tori Baker, and I'm here in Cordova with the University of Alaska Fairbanks with their Sea Grant program. And I have the pleasure today of introducing Bree, who is also a research faculty with the Marine Advisory Program, and she's out of Kodiak. And Bree, this is your presentation, but a lot of people here also know Kate Wynn from many years ago. And so Kate Wynn and Bree are very, very close colleagues. Kate's also in the Marine Advisory Program. So she's going to give us a tour today of some of the amazing work that she and many colleagues have been doing over many years around Kodiak with all of the different whales. And I guess not with too much further ado, Bree just got off a cruise with Jan Straley and others off of Dave Jenkins Auckland. So she's got her land legs back, and you probably saw her running all over town today, literally. One of her other claims to fame is she is an endurance athlete and a marathon runner, and someone who we always appreciate having at a meeting because she makes us get up and move around and like get our blood and circulating. So again, Bree, thanks for taking the time on the end of your cruise to be with us.
Yeah, thank you for having me, and thanks to Tori for kind of pulling this together in sort of a haphazard manner, the small window of time that I had in town here. So I'm glad that it was able to work out. I'm going to try to keep this as casual as possible, so please feel free to interrupt and ask questions, or if you want clarification on anything, feel free to jump in. As Tori said, I work out of Kodiak with the Marine Advisory Program. So what I'm going to talk to you guys today about, let's see if we can make this thing work first of all. Nope.
It was working earlier.
It was working earlier, I saw it.
Yeah, if you click it, click this one.
Click that one, that's the one I clicked. Well, that's okay. So what I'm going to talk today is primarily about a project called the GAP project, which I'll describe in a little bit more detail, and then how whales fit into this GAP framework, the methods and tools that we use to study the whales around Kodiak, what we've learned so far, and then where we hope to take that next in the next couple of years. Really?
Yeah, I'll do it.
There we go. So GAP stands for Gulf Apex Predator Prey Project. It was initiated in 1999 through government appropriation to the University of Alaska Fairbanks, to Kate Wynn and a couple of other faculty that were in Kodiak at the time. And it was funded in part in response to the precipitous decline of stellar sea lions, which many of you are probably familiar with. But there was a dramatic decline in the western stock of the stellar sea lion population beginning in about the 1970s. And so GAP was, well, now we need to go back. Striking out here. There we go. So this is the Kodiak archipelago, and so GAP sought to answer questions that were of immediate biological and economic concern that were triggered by these stellar sea lion declines. And so we looked at a number of different components of the ecosystem, including predators such as sea lions, whales, predatory fish, and then other indicators such as the piscivorous prey populations, all the way down to the zooplankton, as well as a number of marine bird species as well. And in the early years of GAP, we had every component covered. We had researchers working on sharks and the larger fish populations, zooplankton, birds, arrowtooth flounder, pollock. We had a number of different projects, and over the years, as people have sort of spread out and our money has dwindled, we've kind of pared things down a little bit, and now the primary focus of GAP is on whales and stellar sea lions. So why are whales a component of the GAP project? Well, whales are considered apex predators, and they're significant consumers in any of the marine ecosystems where they live. Their diets are often closely linked or even directly overlap with other predator diets, as well as commercial fisheries. And the consumption, it's modeled sometimes that it can actually exceed the removal caused by commercial fisheries. So they are a significant component in any ecosystem. The other component is that most large whale populations were severely depleted by commercial whaling throughout the 18th, 19th, and 20th centuries. And so removing this massive biomass from an ecosystem without having any repercussion is kind of naive to think that would happen. So you remove this big biomass and you have what's called a trophic cascade, and the removing of the large whales trickled down and maybe had impacts all the way down to the lowest trophic levels. In turn, in most recent years, since these whale populations have been granted protection from commercial whaling, they're all now doing very, very well. And so you kind of have this reverse effect now. The removal of this massive biomass and now the influx of this massive biomass. And so there's a lot of complex interactions that can happen when you toy with the ecosystem in that way. And finally, this recovery of the whale populations is occurring in a very unique time. We're seeing environmental change that has been previously undocumented, and so it becomes very important to understand the roles that these whales are playing in an ecosystem perspective, so we can try to understand how long-term impacts on their predation is going to impact the ecosystem, but also how this climate change is going to impact them. So around the Kodiak archipelago, we focus our efforts primarily on 3 species, humpback whales, fin whales, and gray whales. And so our research within the GAP framework focuses on the distribution. So where do we find these whales and when do we find these whales? The abundance, how many are there? Foraging is another huge component. So what are they eating, when are they eating it, and how are they eating it?
So is this bubble feeding that's happening?
That is not bubble net feeding. That is just a really tight group of humpback whales. Humpback whales in Kodiak do not do that social bubble net feeding that humpbacks are kind of infamous for. We don't observe that there. So the methods and tools that we employ to study these whales, to do the distribution and abundance, we do photo identification, and we also do aerial surveys. And for the foraging ecology components, with respect to the gray whales, we do mud grabs, and we also sample their fecal plumes. And for fin and humpback whales, we do tagging and prey assessment studies, as well as implement stable isotope ecology. I'm going to talk about each of these in depth, so that's just an outline of what we do. And long-term studies, if we continue to do these focus studies over several years and even decades, they really help us to understand the trends in abundance and differences in feeding behaviors between these different whales and how they fit into the bigger picture. So distribution and abundance, the primary tool we use is photo identification. And as many of you probably know, each species of whale has some feature that is unique to individuals. So humpback whales, it's their tail flukes. Fin whales, it's their dorsal fins. Gray whales, it's their dorsal flank, and sometimes their tail flukes, they don't fluke up as often as humpback whales do. So we take these photographs, and by repeated photographs of individual whales, you can start to understand how many individual animals you're dealing with. You can come up with an estimation of abundance. So we can document what's called site fidelity. So how often is an individual whale returning to the same spot to feed every single year? Seasonal occurrence, how long are they in the area feeding? The migration routes, most of these species that we deal with are migratory, so we can, by collaborating with other researchers in other parts of the Pacific, we can start to come up with some connectivity between these migration corridors. And then finally, the longer that you do it, the more you learn about the age and sex structure of the population. So with respect to the humpback whales, as I said, they're identified by their tail flukes. And to date, we have almost 1,300 individuals documented within the Kodiak region. And that's through 2012, so that doesn't include the most recent years of effort, this last summer's worth of effort. We have an average resite rate, which means the number of individuals that we see that have been previously seen. We call that resighting, and it's at about 30% for the Kodiak area, which is actually mid to low for other humpback whale populations. I know locally the resighting rate is actually quite a bit higher, and in Southeast Alaska it's quite a bit higher. So that indicates that maybe we're dealing with more animals. We have a larger population. We've been doing this long enough now that we're actually starting to resight calves as adults now, and previous calves with their calves of their own now. So that's another advantage of this long-term study, is you can start to look at things like calving intervals and pregnancy rates. Several years ago now, it doesn't seem like it's been that long, but there was a North Pacific-wide study of humpback whales, and it was a very big matching effort of photographs between all known feeding and breeding areas in the North Pacific Basin. And from that, we got a little bit better picture of some of these major migratory routes. And so for the Kodiak animals, we saw matches to all 4 major breeding areas for the humpback whale in North Pacific. And while most of them went to Hawaii, we had 95 individuals matched to Hawaii. There was actually a fair number that went to both of the Mexico migratory destinations, both the offshore and the mainland Mexico breeding grounds. And then we had this one guy go all the way to Japan. So unlike Southeast Alaska, which is pretty much a high 90% rate to the Hawaiian Islands, we seem to be a little bit more diverse with where our whales are headed.
When you said a guy in Japan, it was a male.
Oh, I don't know.
Oh.
That's a generic guy kind of thing. Yeah, no, I'm not sure what the sex of the animal was.
What percent of these matches of the 1,288? So, about what percent are we seeing?
Well, it's a little bit of a misconception here because this matching here was within that suite of that project. So this project that I was talking about was called Splash. It took place between 2004 and 2006. So it was only the photographs from that chunk of time. So you're probably talking about a few hundred animals over that period of time. So it's actually a pretty high match rate to the breeding grounds. So fin whales, their distinguishing feature, unfortunately, is their dorsal fin. They don't fluke up. Every once in a great while, you'll see a fin whale fluke up, but very, very rare. So we use their dorsal fin shape, the angle, and sometimes they have little holes or pockmarks and nicks and things in their dorsal fins. But it's very difficult to identify these guys. I've picked out ones that are obvious, have obvious features on them, but most of them are very clean and don't have much to go on. And so they require. I see your question. Just say it. They require an almost perfectly perpendicular, beautiful light, calm seas, wonderful picture of these animals. So it's been much more challenging for us to build our photo identification catalog. But we're up to over about 200 individuals now, and we're starting to get the hang of it, starting to get better at it, and in building into some of those resites that we see for the humpbacks. What was your question?
Why are you not taking pictures of the chevron?
You don't see it very often. I don't know if it's an issue of water quality or what, but yeah, I know on the East Coast, oftentimes they take a picture of the chevron, which is a sort of a white blaze pattern that happens on their rostrum and into the back dorsal area, but we just don't see them. Yeah. Yep.
That's the main characteristic that's used in East.
Yeah, I wish we could. I really do.
So,
yeah.
So this is how we identify minkies is by dorsal fin.
Which is probably very difficult as well. Yeah. Okay, go ahead to the next one. And then the gray whales, finally. They will fluke up every so often, so we try to snap a fluke photograph when we see them. But primarily, you use this dorsal flank for these animals. Gray whales don't actually have a dorsal fin. It's kind of this little hump followed by these ridges here. So we've only done 3 dedicated cruises to photograph gray whales, and we generally get around 30 individuals that we can photo identify within that week-long window or so that we're focusing on these. Interestingly though, in each of those 3 years, 2008, 2010, and 2012, none of the same individuals were seen in any of those trips, between the trips. We did match to the catalog, which is called the Pacific Coast Feeding Group of gray whales, and so the Pacific Coast Feeding Group of gray whales are animals that are choosing to not follow their entire migration route up into the Bering Sea. Most gray whales will migrate from Mexico along the Pacific coast and into the Bering Sea. There are a handful of pockets along the west coast where there are feeding gray whales year-round, and so 4 of our Kodiak whales were sighted in other regions of these Pacific coast feeding groups. In British Columbia, Washington, and Northern California. Yeah.
Do you preference left or right side for those photos?
You try to get both if you can, but I think they prefer right sides.
They're right though.
Yeah. They're tricky. They're really tricky as well. Unfortunately, we collect the photos and then we send them down to someone who maintains the entire gray whale catalog in Washington. So we don't have that burden of matching the gray whales. So this is a chart of Kodiak here showing this is all of our aerial sightings, and this has been between 1997 and then this is up to the most recent survey that we did just last month. So on this chart, the gray whales are the green dots, fin whales are the blue dots, and red are the humpback whales. And so you can see there's an obvious gray whale habitat up here on the east side of the island here. This is called Kodiak Bay, and so that is where the majority of gray whales can be found. And the other thing that is really striking to me about this is that while there's definitely some overlap between fins and humpbacks, you can see that the humpbacks tend definitely more towards the east side of the island, and the fin whales tend more towards the west side of the island. So there's some habitat separation going on there for sure. So go ahead. So this is the results of the average number of whales that we see by month during these aerial surveys. So this again is 16 years or so of aerial survey data and the number of animals that we see on average. Colors are the same, so you have green is the gray whale, blue is the fin whale, and red is the humpback. And you can see we see whales year round. There's obvious seasonal peaks that happen kind of when you would predict in the summer, like August, August and September, but it's really, there's not a real dramatic like high peak season for whale abundance in Kodiak. The other thing is we do have feeding gray whales year round in Kodiak as well. So looking specifically then next at the foraging ecology of the gray whales, we go into that area of Kodiak Bay where they all are found, and we do the photo identification, but we've also done these mud grabs. We use what's called a Van Veen grab, and you just drop it to the bottom and take a big scoop of mud and sift through that. And what we've been finding is these little guys down here, which are a zooplankton called Cumacean, and they're generally not considered to be that great of prey, but they are so dense in this area. There's some of these Van Veen grabs revealed the highest densities of Cumaceans anyone's ever documented. So clearly it's enough for the gray whales, because when we sample their fecal plumes, all we find are little Cumacean shells. So that's been pretty consistent over time as well.
So how do
Do the
they
gray whales have to dig them up in the mud? Or?
They don't have to. You do see some gray whales feeding pelagically in the water column, but here and most of the time we call gray whales mudsuckers, because they do kind of lay on their side and dig these channels in the mud. And so oftentimes you'll see these plumes coming out, especially when we're doing these aerial surveys. You'll see these plumes coming out, and you're not sure which end it's coming out of, because they'll sieve the mud out as well from their mouths. Was that?
I was going to ask.
Yeah. Okay. So then, with respect to the fin and humpback whales, we try to tease out some of their foraging behavior using tagging technology. And in the early years of tagging, we used this crossbow method here. So this is an example of our tag, the early tags. This is a suction cup, and then we would fire it with a crossbow. This here is a time depth unit, so it would relay the time back to us, and then it has a little VHF. So when the tag comes to the surface, it gives us a little beep, and we can find either the tag if it's fallen off, or we can locate the whale. Go ahead to the next one. So this is just a series of photos to let you know how easy it is to tag a whale with a crossbow. There, in case, go ahead to the next one, in case you needed to notice, that's the tag there, and that's still the whale over there. So it wasn't all that effective. We had a number of very, very frustrating attempts to do that. This is my personal favorite because it has a suction cup but no tag. Don't really know what happened there. So the next thing is we decided we wanted to be better at tagging whales. So we began this process of improving the whole tag design, and we've moved from this crossbow launching a square tag into basically what's a modified potato gun. So it uses compressed air, and it fires a cylindrical tag rather than that block. And we still use the suction cup design because our tags are short term. We just want a couple of hours of data, not the long-term satellite type tags. So we're now using this pneumatic launcher, and then our tags are made out of, it's just aluminum actually, and then the components slide into that aluminum housing. and we've increased our success rate dramatically by doing this. So it's just a suite of some of our tags attached to the various fins and humpbacks over the years. Go ahead. So what we do is the type of tags that we use, they're not archival tags. So that means you don't have to get the tag back to get the data. It actually relays the signal to a hydrophone on our boat in real time. And so you have that data in real time, and you can follow what this whale is doing as it's doing it. So we've managed to tag 10 humpback whales and 4 fin whales, and so this is just a brief summary of some of the major dive parameters that we calculate based on this dive data. And it's not, the details are not terribly important. What I want you to take away from this here is that fin whales seem to be kind of better at everything. They go deeper, they spend longer time at the bottom, they do spend a little bit longer time recovering, they cover more distance when they're underwater. The only thing that humpbacks are better is that their entire dive time is a little bit longer. But what is particularly interesting to me is this efficiency parameter, which we interpret as, so you have a certain 5 minutes dive time, for example. How much of that 5 minutes are you using to feed versus coming up and down or resting at the surface? And so the fin whales actually have a little bit higher efficiency parameter. They're not significant differences because we have small sample sizes, but I think it's interesting to kind of keep these things in mind as you go forward and you start planning additional studies and trying to figure out the feeding ecology of these animals. Okay, so at the same time that we're doing this tagging study, we have a larger boat with us that's doing prey assessment. And so what we do is a hydroacoustic assessment of the backscattering layer. And we use 2 frequencies, a 38 kilohertz, which lets us see the fish in the water column, and then a zooplankton frequency of 120 kilohertz, which lets us see the little critters. And then we try to drag a net through what we see when we can do that. So go ahead there. So we get all this backscatter data, and when there's a picture of a fin whale fluke, by the way. And we take it by year or by survey, and we try to look at it visually. So what is the screen showing us in real time? What are we looking at? And then we do a technique called multi-frequency differencing, which I'll talk about in a second. And then we try to do a vertical correlation to the depth of dive that tag is feeding back to us. And then we look at it during daylight hours only, because when the sun goes down, the prey field kind of goes and just blows up, so it doesn't really help us. So this multi-frequency differentiating strategy, what it does is it takes your picture of your backscatter and it basically splits it up. So since we use 2 frequencies, the fish frequency and the zooplankton frequency, can take that picture and it can say, okay, this much of that backscatter is fish and this much of that backscatter is zooplankton. So what we can do is, that's fine. No, it's fine. So each year we can look at our picture and we can say, okay, what is the ratio of fish that year versus to the amount of zooplankton? So it lets us have a relative measure of how much of each of those prey categories is in a given year. So the next several slides are going to look like this, and I'm just going to go year by year each survey that we've done so far where we've had the tagging and the prey assessment at the same time. And what we'll have here is the frame on the left is the 38 or the fish sign. The one on the right is the zooplankton sign. The black bar will indicate where the whales were diving, the average depth that the whales were diving. And then the little pictures of the whales over there will indicate the relative amount of each species that we saw. So that's going to kind of be the theme for the next several slides. So this was 2004. It was a year that we saw a lot of humpback whales and just very few fin whales, just a smattering of fin whales. And when we tagged humpback whales and correlated their dive depth with where the prey was found, we found that they were correlated to where the fish sign was and not to where the zooplankton was. And even just looking at the picture here, you can see these big red balls of fish. That's real typical of a fish sign. And you don't see the fuzziness that you would see of more typical of zooplankton. So when we did that fancy multi-frequency differencing technique, we found that the overall ratio favored fish. There was almost 2 times as much fish backscatter than there was zooplankton. And that year we were able to sample and we found that the little fish were capelin. And then we moved to 2005 and we see a very, very similar picture. You see balls of fish again showing up. You start to see a little bit of some fuzziness kind of showing up in that zooplankton picture there. So there was a little bit more zooplankton that year, but we saw similar numbers of humpback whales, just a few fin whales around. Again, we tagged humpback whales and saw that significant correlation to the fish sign and not to the zooplankton. And the backscatter could be attributed more to fish than to zooplankton, but less so than in 2004. And again, the fish showed to be capelin as in 2004. So then we jump ahead to the summer of 2007, and this was a year where that is a completely different picture. There's hardly any fish showing up for us at all, but you have this on the zooplankton frequency there. It's just packed. We saw, I think it ended up being like 60 meters thick in some spots of this massive dense zooplankton school, and it was we saw more fins and humpback whales in one place than I've ever seen. It was absolutely incredible. And so not surprisingly, when we got tags on these animals, they were targeting that very, very dense zooplankton layer, and there was no fish really for them to target. So and that multi-frequency differencing showed that there was far more zooplankton than fish. And then we did a little bit of net sampling, and we attributed that zooplankton backscatter to euphausids or to krill. Then we were fortunate enough to do a winter trip in that year in 2007. And we found an interesting story here. We went to an area where there's overwintering herring, and there were a few fin whales and a few humpbacks around, not huge numbers, but enough to work with. And we actually were fortunate enough this year to get tags on both species. So it was the first time that we were able to document the feeding behavior of the two species at the same time. And we found that they were actually targeting the zooplankton layer, which you can see in the valleys here. So the big red blob would be a school of herring, and then the zooplankton is in these valleys. And we did the correlation analysis, and it showed that the whales were diving to depths that were consistent with the zooplankton layer and not with those schooling fish. Surprisingly, though, the multi-frequency differencing attributed more of the backscatter to herring than to the zooplankton. But we're talking big, massive schools of herring, and so I guess it's not that surprising really when you think about it. But I think what was surprising to us is that the whales weren't targeting the herring. But when we did net sampling on these herring, they were huge, and so it may have just been too much, too big. The fish were too big for the whales to forage on, and the mix of zooplankton was kind of a little soup of everything: euphausids and copepods and chaetognathes.
So, go back. You were saying it was large herring, 25 centimeters, but
that's pretty big for a whale.
That's big for a humpback to go through its.
Yeah, generally they say that humpbacks prefer prey that's less than 30 centimeters, so that's getting up there for them.
Their gullets about the size
Yeah,
of a grapefruit.
Yeah, their gullets are quite small, which is always surprising to folks. So, and honestly, I don't think the whales around Kodiak are very good at catching herring. There are not very many populations of herring around Kodiak, and so they're not skilled at it. And so then we go back to 2009, and we see sort of another picture altogether. We saw very few humpback whales and quite a few fin whales. So it was kind of reverse of what we had seen in the 2004, 5, and 7 summers. But looking at the picture of the backscatter there, you can see that in general it was pretty sparse. There wasn't hardly any fish showing up. We saw a pretty weak zooplankton layer there. We did tag fin whales, and they were correlated to the zooplankton layer pretty strongly. Interestingly, though, there was more. There ended up being more of the backscatter attributed to fish, but it was mostly large pollock that were the fish sign in this case, and those are generally not, adult pollock are generally not considered whale prey. So there should be you have
Just a
questions?
question,
Yep.
on each of the summer cruises, were they fairly close in the dates, or were they all
Yep,
over the same?
August in the same bay of every year. Yep.
Were any done in the dark?
Nope. No, we truncate all our data to daylight hours. So in 2011, we kind of reverted back to this pattern that we'd seen in 2004 and 2005. That zooplankton kind of disappeared, and we see the return of these balls of capelin. And again, we see a ton of humpback whales and only one fin whale hanging out. I think he was a little confused. And see the black bar correlated to the fish layer, and not so much to the zooplankton. And the fish swamped basically any zooplankton backscatter that we were seeing that year. Go ahead. Okay, so the next tool that we use in studying this feeding behavior are stable isotopes, and I'm going to give you the briefest primer on stable isotopes. So basically, stable carbon and nitrogen are what we use, and they reflect the ratios of carbon and nitrogen in whatever prey has been consumed. So you can follow the ratios of carbon and nitrogen up the food chain, basically. Now, the carbon signature remains fairly constant as you move up the food chain. So or the food web trophic level. So it allows us to pinpoint where the animals were feeding. It gives us an idea of feeding location. Nitrogen actually increases as you move up the trophic level. So the higher the ratio of nitrogen, the higher that animal is feeding trophically. So it gives us an idea of where they're feeding. So we do species comparisons, but then we've also done some diet modeling specifically with the humpbacks. You can go ahead and switch again if that works for you. So how we do this is we collect skin samples from whales using a modified rifle. It just takes a little sample. It's about the diameter of a pencil eraser and a little bit longer. And we use the skin from these samples, and so far we have almost 350 humpbacks and 22 fin whales sampled, but not all of those have been analyzed for these carbon and nitrogen ratios. But we have quite a bit done so far. Go ahead. So this is looking at what we've seen between the two species. So the x-axis here is carbon. So remember, that tells you where the animal is feeding. And generally, if it's a more negative value, it's been feeding pelagically or offshore. And as you get more towards the less negative end, it's more of a benthic or a coastal feeding behavior. And then, as I mentioned, as you get higher on the nitrogen scale here on the y-axis, that means the animals are feeding higher trophically. So just the snapshot here, you can see fin whales are feeding lower trophically and more pelagically. Humpback whales are feeding higher trophically and more coastal. Now, looking specifically at the humpback whale diet, so what we did here is we took samples from humpback whales from 2004, 2005, and 2006, as well as samples of all possible prey that they could be eating. And we threw them into a model, basically, that looks at these values and tells us how much of each prey the whales could be consuming. So I kind of have broken it up. There's a little bit of animation, so we can go through that. So we have two groups. What I'm grouping together as the purple triangles there are little fish. So that's Pacific sand lance, capelin of a couple age classes, and age 0 pollock. And then big fish are the couple age classes of eulicon, the age 1 pollock, as well as Pacific herring. And then we have krill down here as the yellow triangle. And that's just that same plot that I showed. You have the carbon signature on the x-axis and the nitrogen signature on the y-axis. And it kind of follows what you would expect. You have krill, who are sort of at the bottom of that food web there. So they're feeding very low trophically, and they're feeding, they're actually feeding more pelagically. And so that's a very good trophic picture of what you would expect between the whales and the prey. And the blue circles are the whales, so they kind of tend up towards that big fish area. So like I said, you plug all this into a model and it spits out how much of each of these different groups the whales could be eating. And what we found out, along here we have each of the years, 2004, 2005, 6, and then all the samples combined. And this would be the proportion of their diet that was comprised of each of those different categories. So the orangey yellow color is the krill, the purple colors are the little fish, and the green colors are the big fish. Go ahead and click there. Well, that's what I just said. Go back one, just real quick. So what we found is that some of these bigger species of fish, they never comprise more than 1% of the diet. Whereas most of the diets were made up of these littler fish and then the krill. So we went ahead and we took all those big fish out of the model and reran it to see what would happen. And what we saw is, go ahead and click again, yeah. We saw a decrease in the importance of the zooplankton or the krill species and an increase in the importance of the fish species. And this is just if I grouped all the little fish together. So you can see, like in 2004, most of the diets of those animals was comprised of the fish species, where it was a little bit more equal, one third to 2/3 in the other 2 years, and then when you consider all the humpback whales combined. So what that tells us is that around Kodiak anyway, there seems to be very little or even no consumption of herring, eulachon, and age 1 pollock. But they seem to be pretty focused on capelin, juvenile pollock, age 0 pollock, Pacific salance, and then krill. And that there does definitely seem to be some annual variability. In 2004, they definitely were more focused on the fish, and in 2005 and 2006, they took some time to munch on krill. So what does this tell us so far? Well, all species, the gray whale, humpback whale, and fin whale, are seen around Kodiak year-round. And the populations all seem to be either stable or increasing. The long-term photo identification that we've done tells us quite a bit about their seasonality and also about site fidelity to individual feeding areas around Kodiak. Gray whales seem to have a very restricted habitat, that area off of Ugak Island or Ugak Bay, and are definitely specializing on those Comasian zooplankton. So it's not likely that they're really competing with prey or habitat with the humpbacks or the fin whales. And then looking at a very broad picture, fin whales are west siders, humpbacks are east siders, and there's a couple areas where they intermingle, but there is definitely some habitat partitioning happening between fin whales and humpbacks.
Do you have any idea why the fin whales prefer the west side of the island?
I don't. I can tell you that they're starting to slowly creep around to the to the east side. I mean, it could be just a prey choice. It could be a niche separation thing where they don't, they're trying to do their own thing over here, and humpbacks are trying to do their own thing over there.
Yeah, herring abundance numbers for the east versus west, because all the herring I've ever done around Kodiak is all on the Shelikof side.
Most of the herring, there's some herring on the east side. In fact, in that bay where the gray whales hang out, up in the head of the bay, there's herring on that side. But there's not a ton of herring around Kodiak in general. So combining that tagging data with the stable isotope data, we can kind of think of fin whales as more zooplankton specialists. They definitely are more focused on the zooplankton, whereas humpback whales probably prefer to target fish, but they will target zooplankton when the densities are high enough. But we started to wonder a little bit, and speculating and hoping to go forward with this question, is that are fin whales more efficient foragers, and that they require less dense prey and can do more with less time when they are actually foraging. But in general, because of their higher fish consumption around Kodiak, humpback whales are more likely to have direct or indirect links to other predator diets and fisheries. So real briefly, I just wanted to touch on where we're going in what we've done and since a lot of this work in the last 2 years. We've basically established 3 index sites throughout the island, and we have set up acoustic grids and oceanographic sampling stations so that we can start to get an idea of annual differences when it's a more structured survey design. And then, so we go to these locations whether there's whales there or not. If there are whales there, great. If there's not whales there, then hopefully the data that we're collecting can help us to try to understand why they're not there. Go ahead. So in 2012, we cited during these three different index sites, we cited about 150 humpback whales and about 40 fin whales. We did 250 nautical miles of transecting and 69 CTDs, which are conductivity time or temperature depth profiles of the water column. And we saw regionally high densities of fish or euphausids. So we never saw dense fish and euphausids in the same place. In 2013, we did the exact same transect, so we covered the same distance, did the same CTDs, but we saw much fewer animals. We only saw about 30 humpback whales and 5 fin whales, so it was a very different year. And we saw very little sign, fish or zooplankton sign the entire time that we surveyed. So there was definitely something different happening in 2013, which really just stresses the importance of doing these repeated surveys on an annual basis so that we can really start to understand interannual patterns and what animals are cueing in on and what is important to them. Phew, and that's it.